Hello everybody, I'm Claire and today I'm with my good friend Remy, who's going to show us all about how to extract DNA from fruit. So it's great for me not to be behind the camera for any of this or behind the computer screen. I actually get to be in front, having been the technical support for all of this. I work at Swansea University as a researcher and I look at antifungal resistance. And today we're with Dr. Remy Zarlot and he shall tell you a lot more about himself.
Hello, thank you everyone for attending today's live genomic DNA extraction. I work with Dr. Claire Price at Swansea University Medical School and I am interested in a few problems that are caused by bacteria that are pathogen, and we are trying to investigate them so we can try to find new way to fight those bacteria. I would like to thank Claire because she's the organizer of this festival that we are attending today. So big thank you to you, Claire. Yeah, let's get cracking.
Oh, good.
So today we are in my kitchen and we are going to extract genomic DNA from fruits.
Hang on now, Remy.
Yes.
Before we start, what is DNA?
So DNA stands for deoxyribonucleic acid. So this is a complicated name, but actually DNA is the molecule that contains all the genetic information that make you who you are and what you look like.
Okay.
For example, I have blue eyes. There is some sort of code in the DNA molecule that says that my eyes are blue. And this is this information, this code that is present in the DNA molecule is actually encoded in what we call genes.
What? So are you talking about genes like Levi's and Wrangler?
No. So genes really are little short sections in the DNA molecule that are actually, they actually tell what the cells are going to do.
Okay.
For example, some cells are going to be told to be the eyes, some cells are going to be told to be part of your skin or even your hair. All of what we are and what we look like is actually a translation of all the code that is present in that big DNA molecule.
Wow.
Every organism has DNA in their cells. It's the case for us humans, but also our pets, all the animals, and also, of course, all the plants. It's very easy to extract DNA from fruits. So I would like to show you how you can extract DNA from bananas and strawberries. And I think it would be cool for you to see what DNA look like. Claire, would you like to see what we need for this DNA extraction?
Yeah, because I really don't believe that you can extract DNA in your kitchen.
Yeah, actually, I'm in my kitchen, and everything we need is going to be behind me. So I'm going to switch you around over camera so you can see everything that's going to go on.
Okay.
This way. And of course, we're going to start with the fruits we are going to extract DNA from. So you have strawberries and banana. And I've tested them earlier today and they are very good. I think it's a good season of the good time of the year to eat those fruits. Then we will need some clean glasses, a little bit of cutlery so that we can cut the fruits, some table salt, washing up liquid, some resealable zip bag, and some coffee filters. If you do not have coffee filters, you can also use napkins, paper napkins, and we will see how we can use that later on. So for all the experiment, I'm going to try to set you here so that everything can be seen clearly. If you don't see very well, please let me know so I can change the point of view.
Okay, but I think you've forgotten something, Remy. What else do you need?
Yes, indeed, I have forgotten something, and I need to go to my freezer for this. There is a very important thing that we need that is in my freezer, and it's going to be isopropyl alcohol. So this is a cleaning product that you can buy online. This is necessary that you have isopropyl alcohol for that genomic DNA extraction. There is an alternative to that you can buy at the pharmacy, and I never remember the name of that product in the UK. What is
methylated spirits.
Yes, thank you very much. The isopropanol you will need to extract this genomic DNA needs to be very cold. That's why I can put it back in the freezer now.
Okay.
Okay, so I think we can start. So the first thing we are going to need to do is actually cut the fruits so we have them ready. So, okay, bring this closer. Okay, so what I want to do is just cut the strawberries in smaller pieces for now.
Okay. So you take off the green bit and cut up everything else?
Yes. When I started for today's experiment, I think I had 5 strawberries. That's a good amount to have good extraction from there. And they are not the biggest of the strawberries, but just normal ones. So at the moment, I'm just cutting the strawberry in pieces, as you can see, nothing complicated.
So, because they're quite small, you'd say 5, but if they were a bit bigger, maybe a couple less?
Yeah, probably 2 or 3 would be good.
Okay.
I also have a banana, and I'm going to just cut it in half. And I will take 1/2 of the banana, and I will remove the skin.
See, I've learned a very good skill during lockdown, or the several lockdowns. I've learned how to peel a banana. Do you know how much this has blighted my life for many years, not being able to peel a banana?
So, which way do you start when you peel a banana? By the stem or on the other side?
The stem.
The stem. I need to work on my banana peeling capabilities.
Mm.
Okay, so we have our fruits cut and ready to go. So what we are going to do is basically break up the fruits. Do you know why we do that, Claire?
No, why do we do that then, Remy?
So actually the DNA is inside the cells. It is protected from the outside of the cells by the cell wall and some membranes.
Okay.
So because the DNA is inside the cells, we are going to break the cells so that we can access the DNA which is inside the nucleus. And to do that, we cut those fruits. And after that, I'm going to use a resealable bag. And I'm going to put those fruits in there. Hop. And I'm not going to mix them. So I will do the banana for one and all the strawberries in the other one. If I can manage to open this bag.
That's the hard part, is trying to open the bag.
It is always difficult to open those bags. Okay, so you can see here I have my fruits. Here and here. I'm going to remove the plate. I don't need it anymore. And what I'm going to do is mash the fruits in a sort of a puree.
Okay.
So I really want to break all those fruits to have like as many cells available as possible for the next steps.
All right.
So I just kind of mash them. You can use a spoon, kind of going around to push them, break them down very easily.
That may be easy because you really don't want to break the bag, do you?
No, we don't want to break the bag. It's not a problem if you break the bag, you're just making a mess. But if we can avoid making a mess, that would be good. So
Looks pretty fun.
What I want is really to have kind of homogeneous mixture, so everything should kind of look the same. Okay. I want as little big chunks as possible. think this is a good start. So you can see, oh, I still have big chunks in here. So can you see well what it looks like now? Yes.
Yes, it looks a lot like a puree with some little chunks, but nothing very big in there.
Yeah. So we have in here, and we're going to do the same for the banana. I think the banana usually is much easier.
Yes. And I would suggest if you've got little fingers, or even if you've got big fingers, cut up the banana first and put it in. It might be a little bit easier.
Yeah, you're right. Also, one tip for you, you can see when I'm pressing down, actually the bag is inflating. So you can press on the corner of the bag, and it allows the air to go away. It will make it easier for you.
Ah, no, that is a good tip.
But be careful, because since the bag is open, you may spill a little bit of banana that has been broken down. It really looks like a puree, like a potato puree for the banana. So we really want something which is very homogeneous. So it's all, what would be the word? All the same, all together, anywhere, everywhere?
Yeah, all the same. Yeah, it looks very much all the same. It looks very much all a mush. It doesn't look like there's any chunks in there at all.
I'm going to try again a little bit more for the strawberries to see if I can have them a little bit better. If you do not have strawberries or banana and you would like to try this at home, I have been told that it's very easy to extract DNA from kiwis too. I wonder, Claire, have you ever tried to do that with apples or pears or any other fruit?
No, I haven't. But it would be an interesting experiment to see which give the best DNA amount.
Maybe one day we'll need to try that live.
Yes, maybe. Do you have an apple or pear shirt, or is it just a banana one?
No, I only have a banana shirt. I don't have an apple shirt. But maybe it will be a good opportunity to try to find a nice apple shirt.
Well, yes, of course.
Okay, so I have my mixture in here. What I'm going to do next is to try to do something so that we can even break the cells better.
Okay.
And how we are going to do that is, first we are going to take table salt and put a small spoon in a glass. So you can see my glass in here.
So which spoon are you using, did you say? The small spoon?
Yes.
So you're using a teaspoon?
Teaspoon, yeah. And I am generous with my salt. And what I want is dissolve this salt in warm water. So I'm going to go to the tap and use the tap warm water for this.
So if you decide to try this at home afterwards, while Remy's filling up his glass with water, you don't need to boil any water. You just need warm water from a tap. So don't worry about boiling, just use warm water. And the reason why Remy's using warm water is because it helps the salt to dissolve a lot quicker and a lot better. You can use cold water, but you might be there all day trying to get the salt into it, depending on how much salt and water you've got. I was just explaining, Remy, why you used warm water rather than cold water.
Yes, thank you, Claire. So you can see at the moment that salt is still not dissolved. So we can see it. That is the white substance, and slowly it's going to go in solution and it will not be seen anymore. I'm just mixing it with a small spoon, it's fine. So very slowly we have the salt being dissolved. It is important when you do those experiments that you take your time, because when you take your time, usually it's when it works the best. So there is no need to rush. If you want to do that at home, you can, with the help of an adult. And it's really not complicated. I actually encourage you to do this at home. So you can see now that the salt has been totally dissolved. The water has become clear again. And we are going to put that warm salty water with our mashed fruits. So I'm going to split it in half.
So you had about half a glass of water, so there's about a quarter of a glass of water going into each bag.
Yes, that's correct.
So Remy, what does the salt do? Why are we adding salty water?
Yes, so the DNA molecule is actually a very big molecule. It's actually a very long string. To make it fit into the nucleus, the DNA is actually coiled or looped around some other molecules that we call histones. We use the salt because the DNA interacts with the salt and will untangle itself. So it will become easier for us to extract it and see it. That's why we use some salt warm water that we add to our bag, and when we have that added, we need to mix. So again, I have one corner of the bag which is open, making sure that you can see everything. And I'm going to mix.
That looks really fun, Remy.
It smells nice. I like when I mash those fruits because the smell gets very nice, and especially the strawberries right now are incredibly smelly, and it's very nice. It's a good way to start a Sunday and to prepare for the lunch. Okay, so I'm going to do this with the strawberry, as you can see, and I'm also going to do this with the banana.
So by doing this, making sure the salt gets to all the cells, to all the DNA, is it?
Yes, we really want to have as much contact, and so you need to really mix it well, so it's like very homogeneous once again. So we have all the DNA from all the cells will have access to that salty water and that salt. So we try to get as much as DNA out as possible. So for the next step, we are going to continue to try to break the cells, and for this we are going to use washing up liquid. So I'm going to show you how I do this, and after that we will discuss why it's good. So
okay.
This is just regular washing up liquid. I have my big spoon now that I'm going to use. So this time I'm going to open this and over this bag. I don't know if you can see well. Over this bag, I'm going to just put here some washing up liquid. So a good spoon, that should be enough. One in here. I'm going to do that with the strawberry too. Oh, and I'm making a mess. Okay. 1 and 2. I can't mix using this spoon, it's fine. I'm going to put this in the sink. Okay. And now, again, you are going to want to mix once again so that we really have all of that dishwasher soap being mixed very well. We want to avoid making any bubbles when we do this, but we really want to suspend everything.
Okay. Does it matter if we use too much washing up liquids?
No, I don't think it matters very much. You need to have quite a lot, but I don't think too much is a problem.
So why are we using the washing up liquids?
Yes. The washing up liquid is actually made of molecules that will break down those protective layers that are the membranes that are around the cells. So we actually cut the fruit, mash them, so we broke the cell, and now we are going to break the membranes that are made of that are making that protective layer, so that we can extract the DNA that has been untangled by the salt. So this is how we can prepare ourselves so that we will be able to extract that DNA. So I need to continue mixing. I didn't mix the banana yet. When you break the cells, usually the mixture is becoming clearer, kind of more transparent. When you have like a transparent mixture, you kind of know that you get more, you get good lysis of the cells, so that you know that your DNA will be well extracted afterwards.
Ah.
It is important to take your time to make sure that all the cells are as much broken as we can, that we have mixed very well, so that we get the best results. Okay, so you can see here, this is for the strawberries, and I still have quite some big debris, cell debris and tissue debris. It's like some of the inner core of the strawberries, a white part that is difficult to break. So we want to remove all of those big debris. So the next step is going to be about this, and we are going to work with some coffee filters. So I just get some coffee filters from the shop. I need two, one for each preparation. And what I'm going to do is put those coffee filters over my glass, clean glass. You can use the glass that you used before to resuspend the salt.
Okay.
And I'm going to make a little bit of a filter like that. If you do not have coffee filters at home, you can make your own coffee filter using a paper napkin by just making it like this. You need to be very careful because paper napkins are much more fragile than coffee filters, but you can use this if you do not have any coffee filter.
Okay.
Okay, so and we need to filter our preparation, so one for the strawberries and one for the banana. So I'm going to slowly pour that in. And hopefully we will see at the bottom of the glass, we will have a filtrate that will be where all those big debris will not be there anymore.
Okay, cool.
Doing the same with the banana. And now we need to be patient. So usually the filtering takes a good 10 minutes or so. So maybe it's a good time for you, Claire, to tell us about what you do in the lab.
Okay, thank you, Remy. I do many different things, but one of the things I do work on is something called antifungal resistance. Now, many people have fungal infections throughout their lifetime. Actually, I think all of us will have a fungal infection through our lifetime. And when we do have it, it's usually something like dandruff or athlete's foot or something very itchy. And we never really think of it as being a problem. But now, if you are somebody who is immunocompromised, so your immune system doesn't work very well, so you could be an AIDS patient or a cancer sufferer who is undertaking chemotherapy, then those same fungal infections can become deadly because they can go throughout your body. And every year, more people die of fungal infections worldwide than they do of malaria. Now, we've heard of malaria. We don't really think of fungal infections as being something that's quite deadly. So what I do in the lab is I look at enzymes that are involved in antifungal resistance. So very much the same as what we're seeing with bacteria. These fungi that are deadly are actually becoming resistant to the fungal drugs that are used to treat them. So I look at enzymes, I look at DNAs, I look at the mutations within the DNA to see why these drugs aren't working anymore. So what do you do in the lab then, Remy? You did touch upon it at the beginning, but could you give us a little bit more detail?
Yes, sure. So I actually work with Claire. We work in the same laboratory. And what I am interested in is learning more about those bacteria that cause diseases to humans. And so I work with the bacteria that, the family of bacteria that cause tuberculosis. So in the laboratory, to study those bacteria that cause tuberculosis, we have to extract DNA too. And so even if there is a tuberculosis vaccine in the world, there is still a lot of people that fell ill because of tuberculosis. The bacteria that causes tuberculosis is actually called Mycobacterium tuberculosis. There are around 4,000 genes in this bacteria, and many of those genes we do not know what they do. So in the laboratory, I extract the DNA from those bacteria so that we can investigate those genes, and so that we try to understand what those genes that we don't know what they do actually do in those bacteria. My research is funded by the European Commission, and we have other funders in the UK, but the European Commission is a big funder of research. And overall, I'm very interested in trying to learn more about bacteria and how they are able to grow so that we can develop new ways to fight those bacteria. There is still a lot to learn about any of those pathogens.
Brilliant, Remy. So I'm sorry to interrupt you, but can we go back to the experiment a bit? I see that the liquid that's coming from the strawberries is red. Does that mean our DNA will be red as well?
We will see at the end of the experiment. If any of you have any suggestion about what would be the color of the DNA, you can put a comment in the chat. We will be happy to have any comments in there. I don't want to tell you right away what color will be the DNA.
Okay.
But clearly at this step, we can see that we extract not only DNA, but also other pigments. And the pigments that make the red color, clearly you can see them. And for the banana, you have some yellow and brown pigments that are coming at that phase at the moment.
Okay.
Remember that we are not yet ready to extract DNA, and so at the moment what you have in those solutions is actually a mixture of the various components that are within the cells, not only DNA but also other things such as those pigments. Yes. So this is usually probably the slowest step that we have in this experiment. We need to wait a little bit more, but you can see that I have in here removed quite a few big chunks thanks to the coffee filter. I will try to pick them up and show them on camera. So that's one of the big chunks that there is. And you can see at the very bottom in the glass, we don't have those big chunks anymore. What we want to have is a layer which is going to be around 1 centimeter or 2 centimeters, so probably 1 finger or 2 fingers in here at the bottom so that we can go further. So we are going to have to wait a little bit more at this point.
Okay. So Remy, I saw you put your finger in the filter. What would have happened if we had broken the coffee filter?
If you try to make the things go faster by trying to mix a little bit, this is what I was doing. I'm trying to mix so that it can go a little bit faster. If you're not careful enough, you may break the filter. If you break the filter, it's not a big deal. You will have those big chunks that will come down. And what you will have is just take another clean glass, and after that put a new filter over that clean glass, and you can refilter what you have gone through. So you can save the day by adding another step of filtration, basically.
Ah, so it's not too bad then.
It is not too bad, yes.
We won't want to ruin our experiment, would we, by being not patient enough?
I'm trying to put a little bit more of liquid because the more volume you have on top of the filter, the faster it goes because there is just more weight over to push down this liquid to go through the filter. And we will be there fairly soon. So while this is still going on, maybe I can start preparing for the next step.
Okay.
So what's going to come next is you are going to need 2 clean glasses again. So I'm going to put those on the side. And I'm going to pick 2 clean glasses, put them in front of the camera. This is what we have in here. And I'm going to go and get my isopropanol from the freezer. Okay, so you can see this is what we are going to need, and what we are going to do is put a good 1 centimeter, 1 finger layer of 2 at the bottom of the glass.
So while Remy's doing that, as he said earlier, you don't need to use isopropyl alcohol. You could use some methylated spirits or surgical spirit from the pharmacy. You can also use other alcohols like vodka. This has been done before with this. But what you've got to remember is the alcohol content in a bottle of vodka is quite low. So the isopropanol that Remy's using today is quite pure. It's 99.9%, whereas vodka and that is very much lower than that. So you would have to use quite a lot, much more than 1 centimeter or 1 finger's worth of the alcohol. So it's much cheaper and much easier to buy isopropanol because vodka and that is very expensive, and it's really a waste for your parents if they have to buy lots of it and can't drink it.
I can see that in the comments we have Daisy that said that she thinks that the strawberry DNA will be pale pink and the banana will be brown. So let's have a look. We will have our answer in a few minutes. So you can see that I put a layer of isopropanol at the bottom of the glass. It's between 1 and it's actually 1 finger. So I'm going to just put that on the side, and I'm going to remove the filter from the glass, and I will put it in one of my bags, so I will use that as a trash. Try not to break the filter and try not to put anything inside the glass. This way it will be good.
So everything that Remy's using today can either be recycled or thrown away in the trash. There is nothing that needs any special disposal. So we have a question in, Remy. Where can we get isopropanol from?
So I order isopropanol from Amazon. It is a cleaning product, so if you go to you probably need to go to a specialist for cleaning products, and you will be able to order that.
Yeah, but Amazon sells everything, and it's freely it's available there quite easily.
Okay, I'm going to start with the strawberry. What I'm going to do is hold this isopropanol, the cold isopropanol, at an angle. Let me try to see if I can have a good point of view for the camera.
Okay.
Moving around a little bit in my kitchen, which is not very big, so I need to move things around. Okay, I'm going to be ready to go now. What you want to do is really take 1 glass like this, where you have your isopropanol, and in here, very slowly pour through the isopropanol your extraction. You can see that I'm going very, very, very slowly. And you want to go very slowly because what you want to get are actually be 2 layers at the end, where we have, you can see 2 layers. At the bottom you have your strawberry extraction juice, and the isopropanol is still clear at the top. And you can see that we have some things already forming in the glass. So I'm going to let that rest on this side, and I'm going to do the same for the banana. So again, tilt your cold isopropanol glass and very slowly pour over that your banana filtrate.
So while you're doing that, Remy, we've had a question from Daisy. She said, can you use hand sanitizer instead of isopropanol?
This is a good question. I would say it's likely to work because hand sanitizer is made of probably 60 or 70% alcohol. So I would say this is actually a good alternative. I never thought about doing it this way, but I would say it's probably going to work. If you use hand sanitizer, make sure that you have a wide, like 3 to 4 centimeter layer of that at the bottom of your glass. And with that, you should be able to go.
Good. Actually, Daisy, that might be a good way to do an experiment, is to try hand sanitizer against another alcohol to see which is the best alcohol for DNA extraction.
So let's have a look quickly at what's happening in here. You can see now that we have some white cloud forming, and it's very clearly seen in here with the strawberry precipitate or extraction. And with the banana, it's going to take a little bit longer because I did it afterwards, but you actually have the same thing. And we can see from a profile view, if I focus on the right thing, that you have some big clouds forming inside at the bottom and at the interface between those two layers. So let me have a look at the script to make sure that I'm not missing anything. Oops. Am I still on camera? Yes, okay.
Oh, your camera's gone off, Remy.
Okay, should come back in a second.
Oh, yeah, we're back.
Okay, I made a mistake. I touched the wrong button at the same time. So, what's going on in there is you are actually extracting DNA. The DNA actually prefers to be with the isopropanol rather than staying in that salty water solution. So what's happening is the DNA is going to start forming clumps into something that we can see. Those are the white precipitates that are actually DNA precipitating. So the DNA that you can see is those clouds in here. This is quite a large amount that you can go and fish with a spoon. And this is your DNA that you have extracted from your strawberry. You can see it's like very large amount. And when I pull on it like that, you may be able to see, oh, I need probably to focus on my fingers. Here we go.
Yeah.
It's kind of stringy. You can see strings forming when I pull on this, right?
Yeah, yeah.
So this
Your
is
camera's falling.
this is actually thousands of DNA molecules in there, quite a lot. And look, I need to take care of the camera and everything. Come on. Doesn't want to focus anymore. Here we go. And yeah, so this is the DNA that is extracted. So what is the color of the DNA now? We can see.
It's white. So it's not pink, it's not red, it's white.
Yes. So the DNA that you extract is actually white, and the precipitates that you get are actually white precipitates. And I should be able, it looks like there is less DNA for some reason today in the extraction of the banana, but you have some of those little clouds. The clumps are much smaller, so it's difficult to fish them out, but you will be able to see that you have some clumps forming in there, and you also can extract the DNA from the banana. I'm going to put you back in here again.
Welcome back, Remy. We can see your face again.
Thank you. This is what we do in the laboratory. We have extracted DNA, in this case from fruits of course, as we said before, we are trying to learn about bacteria and fungi in the laboratory. So we need to extract DNA from bacteria and fungi. And we use very similar protocols for that. Yes. So you should be able to see DNA very clearly. You can do this at home. It's very easy to do, as you've seen. So we can extract DNA to study it. I have a few questions for you, Claire, and maybe anybody in the comment can have guessed too.
Okay.
So do you know how many genes there is in Mycobacterium? I think I may have told it before. So the number, yes.
You might have said it at the beginning, but I can't remember.
Yes, so in Mycobacterium, there are 4,000 genes.
Okay.
But we roughly know the function of probably 60% of those genes. So there are still 40% of those 4,000 genes for which we do not know the function. So you can see, as researchers, there is still a lot of work to do to try to understand much better how those bacteria work. Do you know how many genes we have in the human genome?
Oof, I don't know, but we are much bigger than a mycobacteria, so I would say we have lots more.
Lots more, okay. Actually, 6 times more genes. So we have 24,000 genes in the human genome. Yes. What do you think? Do you think we have more or less genes than a banana or a strawberry?
Okay, so my logic was that because we were bigger than Mycobacterium, we would have more. So a strawberry and a banana are smaller than us, so I think they have less.
Well, actually, you're incorrect because a banana has 30,000 genes.
Wow.
And a strawberry has 34,000 genes. So you can see that it's not the number of genes that make the complexity or make how intelligent a species is, because clearly plants have much more genes than, well, not all the plants, but actually the banana plant and the strawberry plants have more genes than humans, and actually also much more genes than some of the other bacteria. Is there anything else you have you would like to talk about? Or if you have any questions in the chat, I think this is a good time to take questions.
So yeah, so if you want to put your questions in the chat, and I will just say while you're writing your questions that if you would like to do this experiment, then this video will be available on YouTube, so you can feel free to watch it and pause it and to do your experiment with Remy. There's also an evaluation form that is pinned to the top of the chat. If you would kindly fill that in so we can know more about what you thought was great about this session, what you think we could do better, and across the whole festival as a whole. And you could be put into a draw to win a £20 Amazon gift voucher. So please fill in. So we have a question from Ro. So the question is, how do all the genes fit inside of a strawberry?
Yes, so actually the principle is the same whether you are a bacteria or a eukaryote, so a human or a plant. The DNA molecule is a very long string of information, and the way you can fit that lots of information is basically to coil the molecule on itself. So the DNA is coiled on itself with the help of different other molecules, notably the proteins that are called the histones. So the DNA is made actually very compact and very small. I think if you take all the molecules in only one cell from a human and you uncoil them up all together and you put them all together on top of one another, the length that you get for only one cell is actually 2 meters in height. It's actually very, very narrow, but it's actually very long.
Wow. So from Daisy, this is more of a statement than a question, but she said, and all these genes are helping in forensic investigations.
Yes, gene can help in the case of forensic investigations. Actually, we don't need to know what contains gene to be able to help with forensic investigation. What you are going to look at is patterns that are contained into the DNA, and you can compare different patterns between different individuals to try to see which pattern is matching what. So what are the chances that one individual is actually involved in something? This is how it works. What we do in the research laboratory is we want to look specifically not at those patterns, but the genes and what are the functions for those genes. And the reason we do that is because there are many genes for which we still don't know what they do, but they might be involved in biological phenomena that are essential for those plants or bacteria or humans to survive. And so if we are able to try to target those essential genes, to try to turn them down or turn them off, we may be able to control, for example, the growth of a bacteria to try to actually kill it. And this is what we do, for example, with drugs that are treating disease treatments, such as antibiotics. Antibiotics are usually molecules that are going to target not genes, but the product of those genes, so that we fight the bacteria and the bacteria is not able to grow.
Brilliant. And we've got another question from Daisy. And it says, is fruit and veg DNA longer than human DNA?
Yes. So I don't remember the details, but to have some space on this genome, to have those genes, you basically need a longer genome. It means that the DNA molecule is actually correlated with the number of genes. And for example, the DNA molecule of Mycobacterium is much smaller than the DNA molecules that we have in our human cells, or actually in the cells of strawberries and banana. There is, to make a gene, to make the information available, you start with 4 bases, so A, T, G, and C. And actually, this is the code that is decoded later on. But you can see that you need 4 bases. So if you want to carry 24,000 genes, the book that would be made of those 4 letters is going to be much longer than the book that would be corresponding to the genome of Mycobacterium. So yes, those DNA molecules of the species that have more genes are going to be longer.
Okay, thank you very much, Remy. I think that was our last question. So all it is now is to say thank you very much, Remy, for that incredible demonstration. Also, thank you to everybody for watching. We're on the last day of the Science Festival, so please enjoy what's left. And don't forget that all the videos, whether you've missed it or not, are all available on our YouTube channel. So please do enjoy. So thank you very much and enjoy the rest of your day.
Thank you very much, everyone, for attending. Thank you very much for asking questions. I hope we have answered all your questions. Remember that you can do that at home. It's quite funny and quite easy to do. You don't need a lot. I think actually using the gel, what was the suggestion as the form of alcohol to use? I forgot the name of the gel.
Sanitizer.
The hand sanitizer is actually a good idea. You can probably try that at home. And yeah, thank you very much. Don't forget that you have more videos to see. You can have a look at the website for the festival. And thank you, Claire, again for organizing this festival.
Thank you. Bye-bye.